Thank you, Chairperson, and also I'm really thankful to the Academy for this kind invitation and allow me to share some of the research activity which is going on in my lab at IACS, and welcome you back to the podium. So, as you could see from the title that today I aim to speak on the domain where we'll be referring to the fluorescent organic nanostructure, and that would be utilized towards sensing a cancer cells by exploiting the oxidoreductase enzymes which are overexpressed in the cancer cells, and we'll take a few examples of that. So prior to moving into that, we'd like to know why you got interested in the organic nanostructures. It's a beautiful manifestations of the self-aggregated amphiphilic molecule, and where from the inspirations came in for that. And the inspirations always come from the nature, as you all know. We try to mimic the several phenomena that's been taking place in the nature, and one of such a simple thing in the nature are the self-assemblies. There are beautiful manifestation examples are there in the nature, starting from this chromatin structures to neuronal cells, and the basic building block of a life is a cell, and which is comprises of a cell membrane, and this cell membrane is one of the finest manifestation of self-aggregations without any forces, outside forces. It's got self-assembled, and because of the presence of this amphiphilic lipidic molecule within that, which is having a hydrophilic head and hydrophobic tail. And to understand the structure functions and the phenomena that has been taking place in the membrane systems, and the scientists and the researchers, they try to make things more simple, and from there the concept came in the mind is the molecular assembly, where we try to design simpler analogs of this lipidic molecule, which can, depending on the temperature, system structures, can give rise to the formation of different manifestations of the self-aggregated structures, like a simple amphiphilic molecule having a hydrophilic head and hydrophobic tail. Depending upon the situation and structure, that can lead to the formation of micelles, reverse micelles, different microemulsions, the vesicles, which are the closest mimic of these membrane systems, and followed by the higher order aggregates like gels, and a very concentrated organic nanostructures like carbon dots. And this is another fantastic manifestation of this self-assembled structure is the organic nanoparticle. So our to go into the today's topic, I just briefly want to mention the research activity in our lab at IACS is centered around these self-assembled organic nanostructures, where we have been working in the development of the microemulsion for boosting the compartmentalized enzymes. We are working in the development of the different vesicular structures as a delivery vehicle target specific. We are working on development of the nanogels, which is nothing but the small hydrogel particle dispersed in the water, which are the huge influences in the environmental remediations and the medicinal chemistry. We also work on the exploitations of the carbon nanotube towards as a cellular transporter by exploiting its hydrophobic surface area. And there are the fantastic another structures which is intrinsically luminescence is the carbon dot, which could be derived from the small organic molecule, and that can be has a huge prospect in theranostic applications. And these fluorescent organic nanoparticles, and which upon aggregation shows the emission, and today I'll be talking about in this domain with a few examples. Now the questions, the fluorescent nanoparticles that are being in sensing applications, and not only sensing, other bioimaging and so on, is there in the literature. So what is new in this fluorescent organic nanoparticle then? Any kind of nanoparticle we are trying to use, or like a semi-quantum dot or organic dye and so on, quite obviously it was generally it is observed that with increasing concentration or upon aggregation, there is a phenomena called the aggregation cost quenching that emit the emissions of quench the emission of these organic dyes. And primarily in most of the cases it happens the pi interactions leads to the effective quenching because of the non-radiative deactivation pathway. So herein, instead of that, if we can make, and also in this quantum dot or metal nanoparticles, they show some sort of toxicity in the cellular system. So instead of that, if we can design an amphiphilic molecule which could be weakly luminescent or non-luminescent, but upon aggregations that can be luminescent, which have a better stability, better compatibility, and better acceptability in the physiological system. So to this concept, in 2001, the Tang group bring in the concept of the aggregation of induced, where a normal molecule which are emissive in solutions and non-emissive in aggregated state, this kind of molecules shown are aggregations in the emission in the aggregated state. And the characteristic feature that was which observed in such molecule is that they have restricted intermolecular rotations, they have charge transfer facilitated by the donor acceptor moiety in that molecule, and they have a generally non-planarity and the chirality in its structures. A few of the examples given over here. So what we plan to make use of this kind of simple molecule naphthalene diamide and naphthalimide in our studies because of two reasons: their abundances, and also your synthesis skill need not to be so challenging. You can make it modulations far more easily using this kind of molecule. So herein we will be using today, showcases today the FNPs in biosensing. So the first molecule which we have chosen is the protein disulfide isomerase. You all know about this molecule, which has been quite abundant and overexpressed in many of the cancer cells, including melanoma, breast cancer, and so on. And PDI exists in two forms, in the oxidized form as well as in the reduced form. And it involves in the proliferation of survival and metastasis of several cancers, participations through the unfolded protein response. And this molecule in the oxidized form available in the endoplasmic reticulum in the cellular systems, which forms a disulfide bond from thiol, and conversely the PDI reduced form, which is mostly available in the endosome and cytosol of a cell, and which converts disulfide to a sulfide corresponding thiol. So this overexpressed PDI we would like to determine by designing this fluorescent organic nanoparticle in such a way that will selectively sense the PDI as well as it can diagnose the cancer cell over the normal cells. So as a result of that, what we have done, we have came up with these two molecules, which is a bit complicated in nature, but after a lot of failure and trial error method, we have come up to this molecule. And you just look at this, it has a disulfide NINSS bond, and it has a thiol bond having a tosyl group connected to the ethylene diamine, which is a ER targeting moiety, this one, and this is a disulfide bond. And it happens to be, we did this corresponding characterization, we found that it shows the self-aggregated organic nanoparticle from the microscopic through the J aggregations, and corresponding fluorescence was observed. And interestingly, upon aggregation, it showed blue emissions for the disulfide molecule in the range of 470 nanometer, and so the bit of yellow emissions around 550 nanometer by the thiol moiety. And we did all other spectroscopic microscopic characterizations. Then we move on to make use of this molecule for the real objective for PDI sensing. So initially what we have done, we have taken this disulfide containing molecule, and this PDI is in its reduced form. What happens? It should reduce this disulfide bond and form this thiol one. So it is a fluorescent one, it is a non-fluorescent one. So as a result of that, what we could see a very easily slow decline in the emission intensity with the increasing concentration of the protein in this system, and that was quite obvious. But what happened, you could see there is little sporadic nature in the declination in the emission intensity. There is some hike, a jump. So why is it so? So looking at the mechanism, what we found, this disulfide is getting reduced by this PDI reduced form and led to the formation of this thiol, and this PDI itself gets converted to its oxidized form. So after a critical concentration, what happened, this PDI and this thiol can get oxidized back to this disulfide by this oxidized PDI. So as a result of that, there is a possibility that this energy, the fluorescence of this disulfide molecule can restore back. As a result of that, there could be a little hike where which did not see the complete steady emission. In natural system, what happens, this kind of catalytic cycles gets ended by a beautiful electron donor molecule like the glutathione or NADPH or DTT and so on. So whatever happened, we have did the same reactions in the presence of the minute amount of glutathione. So since the glutathione has a better affinity towards this oxidized PDI, so instead of reactions with this thiol, the glutathione will start to react with this PDI oxidized form. As a result, this glutathione will be converted to corresponding GSSSG, and PDI will be restored back to its completely reduced form, which was not possible. So eventually this reduced form, this oxidized form no longer react with this thiol which has been formed from the disulfide. So as a result of that, what will happen? We have seen the complete steady decline in the emission intensity in the presence of the glutathione with the limit of detection of the 12.6 to 17.7 nanogram per ml. So the same thing happened when you have done for this thiol moiety which sends the PDI in its oxidized form. But interestingly, this thiol is converted to disulfide, and here you could see there is a drop in the emission intensity of thiol with increasing concentration of the PDI, but there is a beautifully another peak is coming up having a red emission around 625 nanometer, because the disulfide itself also form another aggregated organic nanoparticle which also have a red emission. But here with increasing concentration of the PDI in the oxidized form, you could see again that quenching or enhancing in the emission also not straightforward, because of the similar reason that the thiol gets oxidized to disulfide, and this reduced form of PDI will again react with the disulfide and send it back to here. So here what we have done the same reaction in presence of little bit of glutathione disulfide. As a result of that, instead of reacting on this reduced form will react on this glutathione disulfide and lead to the formation of the PDI in its oxidized form. So PDI on its oxidized form goes back by the presence of the glutathione. So there is easily the PDI in its oxidized form can be sensed by this FONP selectively at a range of 11.7 to 16.5 nanogram per ml. Since so in nutshell, what we it has been done also for the cellular imaging, where we have seen the cellular imaging can be done selectively on the cancer cells over the normal cells. Where the picture is not that so clear, and there is a mean fluorescent intensity of 47 against 189. Similarly, for the oxidized form also can be seen with increasing time and the concentration of the PDI overexpressed in cancer cells. You could see very high rate fluorescence which has been shown because of this formation of the disulfide, which has a 625 nanometer emission. So we have been able to selectively sense the PDI in its reduced and in the oxidized form by the designing of a different kind of FONP. And I will give few more examples of the similar oxidoreductase enzyme which has been overexpressed in the cancer cell, and that also can be sensed by this different aggregated organic nanoparticle. One like the lactate dehydrogenase A, which is overexpressed in cancer cells because it's involved in the glycolysis, and the tyrosinase A, which is also overexpressed in the melanoma cells, which is involved in the oxidation of the polyphenol compounds to the corresponding melanin through the formation of the dopaquinone. So first with the lactate dehydrogenase A, which primarily involved in the glycolysis from pyruvate to lactate formation, because in normal cells the ATP energy production takes place to the mitochondria and TCA cycle, but in cancer cells, because of the high demand of the energy, they bypass this pathway and they move through this glycolytic pathway. In glycolysis pathway, the pyruvate to lactate formation takes place due to the presence of this overexcess of lactate dehydrogenase A. So we wanted to sense this lactate dehydrogenase A by making the diagnosis of the cancer cells. So what we have done, we also had done a lot of permutation combinations, a lot of failures are there, and eventually come up with this molecule, naphthalimide based amphiphilic molecule having a aliphatic pyruvate in its arm. And this molecule, because of the naphthyl substitution, also shows a beautiful yellow emission around 590 nanometer, and it shows corresponding fluorescence. And then we have checked its ability to sense the LDH. So pyruvate to lactate conversions takes place by the presence of this LDH, which is coupled with the conversion of the NADH to NAD+. So when you checked the emissions quenching of this compound FONPs by this enzyme, we have seen there is a decrease in the emission intensity around 590 nanometer. Alongside, there is also decrease in the emission around 420, 425 nanometer, which is the drop in the emission intensity of the NADH. So the question may come then, what is the need of utilizing your FONP for sensing this LDH? Because the NADH itself can do the sensing. But when you follow the sensing mechanism from the Sternberger plot, and the corresponding deductions limit has been determined, we could see our FONP has a limit of deduction around 18.6 nanogram, having a very nice linearity, in contrast to the NADH, which is around 100 nanogram per ml, having a very poor linearity. So there is a 5.4 higher selectivity, as you could see by this FONP for selective detection of the LDH. And that has been also observed in the corresponding sensing of the cancer cells. You could see the normal cells has a very high fluorescence of this FONP having a MFI of 1445, whereas the cancer cells got a 459. So quenching takes place in the cancer cell of the FONP because of the high presence of the LDH. and that was further confirmed by using a corresponding inhibitor of the LDH, which is sodium oxamate, where you could see by the presence of the sodium oxamate there is a restorations of the emissions around 1016 from 459, which makes it more selective that the LDH has been since the FONP, and that was further confirmed by using another experiment where the normal cell HEK has lack of LDH. What we have done, we made a transfections with a construct so that LDH can overexpressed with the GFP tagged, and where we could see there's a lot of green signal, but in contrast the FONP's emissions got quenched because of the high expressions of the LDH in the normal cells. As a result, we have been able to make a selective sensing of the cancer cell or diagnosis of the cancer cell by using these FONPs, and this work was carried out by Kothakoli, Shoumuli and Debolina. And finally, the last one I would like to share that we also have done the fluorometric sensing of the tyrosinase, where we have used this naphthalimase molecule having a morpholinome attachment for targeting melanosomes as well as this catechol moiety, which are selective for the tyrosinase enzyme, and it also showed aggregation induced emission in the green range having around 520 nanometer and so on, and it also showed emission is quenching in presence of the tyrosinase enzyme having a limit of reductions of 58 microunit per ml because of this oxidation of this OH group to corresponding dopaquinone and the corresponding melanin, which shows a very selective sensing, and that selective sensing was also observed in case of melanoma cell in contrast to the other normal cancer cell or normal cells, which are the 1.3 to 3.4 high selectivity. So in summary, I would like to say that we have been able to prepare a different kind of aggregated organic nanoparticle which has been made to be fluorescent, and that has been selectively utilized for diagnosis of different kind of cancer cells by exploiting their AI induced fluorescence property by like making a PDI sensing, tyrosinase sensing or LDH sensing. So in final, I would like to thank all my students, but prior to that I would like to thank my mentor teacher Professor Aurobindo Chaudhary, under whom I have did my doctoral studies, and he had made me a nice guidance throughout my research journey as well as my personal journey too. And I am definitely thankful to all my past scholars who have been instrumental of establishing my laboratory and my research activity at IACS, and of course my current scholars who have been a constant source of learning in my pathway research journey. And thank you for your kind patience.
Thank you, Prashant. A few questions there.
Yeah.
So for this nanoparticle sensing for diagnostics, how many after the onset of cancer, how long would the progression need to take place before you can actually implement the diagnostics? How many cells do you need? How sensitive is it?
Yes, thank you very much. So, Professor Majumdar, I would like to say that what we are sensing here, we are trying to sense taking a cell culture from the cells, where we are taking around 1000 kind of number of cells, and that cells are being able to, because we need a critical amount of the enzymes to be present there, after which we will be able to sense this. If our PDI or LDH concentration is too low, then definitely would not be done. So we need to have at least certain around 500, 700 nanogram to certain microgram ml of enzyme expression is needed, before which we will be able to sense that. So we need a definitely certain level of growth, but I exactly cannot tell you that what exact content and which grade of cancer will be able to sense it. But yes, we need a certain critical concentration definitely.
Very interesting observations.
Thank you.
How do you propose to use this in a clinical situation?
Yeah.
In vitro or on tissue specimens, once biopsy is available, how does one use this?
Thank you, sir. So first of all, our study so far is limited to this 2D cell culture study, but I tried to done this study in the 3-dimensional tumor spheroid model first of all. But unfortunately, the problem is in the tumor spheroid model, in 2D cell cultures, we will be able to segregate out the cells from the dye itself. But in 3-dimensional tumor spheroid model, we are not being able to done that. But it is already intact within the systems. But so it has to be tested in the tissue directly. Instead of 3-dimensional tumor spheres, we have not done that in the 3-dimensional tumor spheroid in the tissue models, but we need to collaborate for doing these studies with the clinical or the hospital samples.
But the tumor is inside the body.
But we need to take a sample. Yeah, but either like the PET scan when we have done, we put a dye inside the samples to make a scanning. So to bring these materials to that level, it will definitely take a lot more time. But for the fast answer, I think the biopsy sample has to be taken out, and on which we should make an attempt to make a sensing over there.
Yeah, we have one last
Sir, I have one quick question.
Yes.
So how effective this method as compared to the laser-based mechanisms such as optical trapping?
I mean, we have been compared this method with respect to the corresponding different other sensing methods. In most of the instances, it is comparable or better. But what method we are referring to that exactly not comparable to this pathway what we are referring to. So in some of the analytical sensing method, it's sometimes better or sometimes comparable to that actually. It's competitive. I would not say it is the only outstanding method. It is competitive.
Thank you. Thank you, Prashant. All the best.
Thank you very
we
much. Thank you.
him from the academy side. Thank you. Our next speaker is... thank you, Prashant. A few questions. If there are.
Yeah.
Yes.
So for this nanoparticle sensing for diagnostics, how many after the onset of cancer, how long would the progression need to take place before you can actually implement the diagnostics? How many cells do you need? How sensitive is it?
Yes, thank you very much. So, Professor Majumdar, I would like to say that what we are sensing here, we are trying to sense taking a cell culture from the cells, where you are taking around 1,000 kind of number of cells, and that cells are being able to, because we need a critical amount of the enzymes to be present there, after which we will be able to sense that. If our PDI or LDHA concentration is too low, then definitely would not be done. So we need to have at least certain around 500, 700 nanogram to certain microgram ml of enzyme expression is needed, before which we will be able to sense that. So we need a definitely certain level of growth, but I exactly cannot tell you what exact content and which grade of cancer will be able to sense it. But we need a certain critical concentration definitely.
Very interesting observations.
Thank you.
How do you propose to use this in a clinical situation?
Yeah.
In vitro or on tissue specimens, once biopsy is available, how does one use this?
Thank you, sir. So first of all, our study so far is limited to this 2D cell culture study, but I tried to done this study in the 3-dimensional tumor spheroid model first of all. But unfortunately, the problem is in the tumor spheroid model, in 2D cell cultures, we will be able to segregate out the cells from the dye itself. But in 3-dimensional tumor spheroid model, we are not being able to done that. But it is already intact within the systems. So it has to be tested in the tissue directly. Instead of 3-dimensional tumor spheres, we have not done that in the 3-dimensional tumor spheroid in the tissue models, but we need to collaborate for doing these studies with the clinical or the hospital samples.
But the tumor is inside the body.
But we need to take a samples. Yeah, but either like the PET scan when we have done, we put a dye inside the samples to make a scanning. So to bring these materials to that level, it will definitely take a lot more time. But for the fast answer, I think the biopsy sample has to be taken out, and on which we should make an attempt to make a sensing over there.
Yeah, we have one last
Sir, I have one quick question.
Yes.
So how effective this method as compared to the laser-based mechanisms such as optical trapping?
I mean, we have been compared this method with respect to the corresponding different other sensing methods. In most of the instances, it is comparable or better. But what method we are referring to that exactly not comparable to this pathway what we are referring to. So in some of the analytical sensing method, it's sometimes better or sometimes comparable to that actually. It's competitive. I would not say it is the only outstanding method. It is competitive.
Thank you. Thank you, Prashant. All the best.
Thank you very much.
Thank
you.
him from the academy side. Thank you.